thanks for the organizer for inviting me to share our work with you. So I really enjoyed the talk from Nicole and Vinod. We gave a lot of really good background information. But in today's talk, I'm going to share with you some of our recent work in using machine learning approach to really study the basic science as well as the clinical implications for psoriasis research. So the high throughput data is actually has significantly advanced biomedicine. For the molecular data, we start with the microarray technology using the hybridization techniques, and to the next generation sequencing, which allows us to not only profile the transcriptomics, but at the same time epigenomics, and more recently about a single cell at the single cell level. And in parallel, for the clinical data, we start with just using some of the demographic variables in our modeling, and then to getting a little bit more structured electronic health records data. So all this kind of big data kind of come into place in the last 10 or so years, helping us to address some of those major questions that we're facing in the biomedicine. That includes identifying factors associated with disease pathology, and how do we understand the biological effects. And then at the same time, this is more basic science research on how do we apply some of those techniques and integrate them to really model the disease process or etiology. Okay, so as we know mentioned about some of the major challenges, I'm just going to recap really briefly. We are dealing with really large number of features with relatively small sample size, especially for molecular data. Just give you a sense for the genetic study that we have been working with. We are easily talking about examining the genetic variations for over 10 million markers in the study. And for those transcriptomics or epigenomic studies, we are dealing with more than 10,000 features. To make things even more complex, so we're dealing with some system that each of those features that only contributing really mild effects. Okay, so we have to really integrate or aggregate multiple different features in order for us to have a more robust modeling. Just as a one example, this is a genetic study that we published a couple of years ago, trying to identify genetic variations with allele frequency difference between the psoriatic patients versus controls. So each dot here representing a genetic variation. So this across the different chromosomes, and the y-axis representing log 10 p-value. Okay, so you see really significant results here, but this could only be achieved when we have enough sample size. Okay, and we have talked about 10,000, 20,000 cases and controls together to obtain these significant results. The reason for that is because we're dealing with, again, really modest effect size, even when we comparing when we identifying a genetic variations associated with psoriasis. So this is it's a breakdown for those top signals that we identified. You can see, except for the really well-known MHC signals, we have over 4 in odd ratios. Most of those are between 1 to 2 in the odd ratio range. And then when you look at the risk allele frequencies in those psoriatic cases and controls, you're seeing really small changes. So that's kind of we reiterate what I was mentioning about, we have to really gather multiple different features in order for us to get a better modeling. So in today's talk, I'm going to focus on 3 different approaches or 3 different research efforts that we have conducted. First one is trying to model the genetically regulated component for gene expressions. We're trying to address 2 of the major things that I mentioned about in reviewing the disease-associated factors, as well as understanding their potential biological effects. And I'm going to also share with you some of those more recent work to try to model the risk for psoriatic arthritis, as well as modeling the drug response using genetic and transcriptomics data. So as I was mentioning about this again, this is the same plot, each dot representing a genetic variation here. The one that's colored are the ones that's significant. The blue ones are the ones that have been established, being identified in peer studies, and then the red one are representing those loci that we recently identified. So identifying this is one thing, but at the same time, we want to know what they could actually do. Okay, and it turns out that majority of those, like over 80% of those psoriasis-associated loci overlapping with the regulatory elements, which means that they are not going to have effect on changing the function of the protein, but in turn they're actually changing the expression levels for the genes that they target. So as an illustration, so this is just cartoon. We have a site that is actually a lot of those psoriasis-associated loci actually overlapping with a site that could be bound by transcription factors. Okay, and the different alleles actually could have different binding affinity. Okay, so for example, there's an individual that carrying homozygous A. Okay, that have a higher binding affinity for these transcription factors, and then in turn it will kind of have a higher expression levels for those genes. Okay. And when comparing with those individuals that only carrying like heterozygous genotypes or homozygous T, okay, in the genetic makeup for that particular marker. So in turn, okay, so this is nice because if we kind of know what this is and what they target, okay, then we potentially know what are the genes that are being influenced by the genetic variations. But there's like again the story is like more complex. First, there could be more than one marker that could influencing the gene expressions. And at the same time, we may be talking about this could be a really tissue specific. So we have to identify a relevant tissue to really model this process. So in the statistical genetic field, there are different computation approaches that have been developed, really trying to model this kind of a genetically regulated component of expression. So what they're trying to do is really trying to break down. Okay, so this is like all the expression, the variations of the expressions that you see in that tissue, and then try to break down to the genetically regulated component, or a component that is just stochastic, or it's just a component that the variations just be explained by because of the changes of the trait. Okay. So so this is the part that we trying to model it. So in order to do that, we have to have the same set of individuals that have both being genotyped as well as having their relevant disease, relevant tissue being profiled for their transcriptome. Okay. So and then once we have that, then for each of those gene expression profile, we could actually apply in this particular study, they apply like elastic lat to try to really select the most informative genetic variations to model the genetic regulated component of the expression. Okay, so you can not only know what are the genetic will have effect on the expression level, but at the same time you can selectively identify those genetic variations that are most informative in the prediction. So once you identify this, okay, you could actually, once you have this like model, then you could do is actually apply on independent data sets, which only may have genotypes data, and then to impute those genetic regulated components of the transcriptome. And then you could actually try to see what are associated with the disease status. For example, in our case, would be psoriasis, psoriatic skin or normal skin. Okay, so just as an example to illustrate one of the challenges, actually we also touch about is like we need to have like some more samples to really have a better estimation. So this is a so we first tried to using our own Michigan psoriatic skin RNA-seq study to try to train the model. Okay, so we have around 161. So what you're seeing here is actually each dot representing one gene. Okay, the performance for each gene. Okay, in the predictions, and then the gray bar representing the confidence interval for the genetic heritability for that gene expression. Okay, and then if we are seeing that the so this like confidence interval, we're seeing all those red dots is actually in the middle of that confidence interval bar that we are seeing about our modelings could capture a majority of the genetic heritability components. So well, you can actually see the really huge confidence interval bar. Okay. When comparing with when we use a larger sample set, okay, to really train the model. So again, this sample set has to have both the genetic data and the expression data. Then we are seeing much narrower, much like more refined confidence interval for the heritability. And then at the same time, we're able to capture a lot of those really good genes for the performance in predicting the expression profiles. Two examples here. So these two genes, we are only getting the genetic profiles and then try to see how are we going to predict the expression levels. And then this is comparing with the observed. Again, the observed could be having multiple different components, but you can actually see how highly correlate for those genes. So these are some of the best examples that we get. And there could be some genes that we may not be able to capture even with really large sample size. So with that, okay, instead we are not looking at a random plot that each dot representing genetic variations. We could actually project it such that each dot now representing a expression or a gene, okay? And then now, in addition to looking at the loci, in terms of the genetic variations, we actually identifying those genes with their expression profiles that are actually regulated by potential psoriasis associated loci that are being differentiated in the psoriatic lesional skin versus normal skin, okay, or psoriatic patients versus control individuals. And then, so in addition to looking into some of those regions that actually also overlap with the previously identified psoriasis loci, we at the same time also identify some new regions, okay, so which give us like some sense that we could potentially improve power. So in addition to using genetic data to really model the expression profiles, we're also interested to see whether there's any potential clinical implications. So that's how, as we also mentioned about like how we could potentially use the genetic data to model the risk of psoriatic arthritis among the psoriatic patients. So both Nicole and Dino mentioned about like some of the rationale why we motivation why we have to do the PSA assessments, especially when we're trying to, if we have a better good screening of those individuals, then we may we may be able to predict it well, then we could have a better screening for those individuals. So this is also a genome-wide association studies trying to associate the genetic variations with the different psoriasis subtypes here. So we actually, again, each dot here representing genetic variation, okay, and we are doing a in this plot we are comparing the PSA versus control, and in here we're comparing with the cutaneous only psoriasis genetic makeup versus the control, okay. And then you can actually see there are a lot of overlap, okay, between them. So it makes things even more subtle if we want to really differentiate those psoriatic individuals that would develop psoriatic arthritis or not, okay. So we apply some nonparametric approach, machine learning approach to really address this. But we first need to make sure that this could be generalizable and robust by breaking the data to training set and test set. Okay, so this is actually reiterate some of what we've discussed, that we need larger sample size in order for us to do that, because we want to make sure that the model that we are going to train is going to be applicable for this data set that we actually just left out and did not use it in the model training. So we break down those into training set and then use tenfold cross-validation to do the model optimizations. And once we optimize the model and apply it to the final data set. And this is how we're going to see how it could apply to the data. So the x-axis representing the top X percentage of the patients that we predict to have psoriatic arthritis. In here, for example, when we are going to look at the top 10% of the psoriatic patients that we predict from the model to have psoriatic arthritis, then we got really high precision. But if you look at the recall rate, then we are actually just talking about like one third of those psoriatic arthritis patients. But if we lose the criteria, Krish saying, well, how about we just like look at the top 20% of the psoriatic patients that we predict to develop psoriatic arthritis in future. Then we got a little bit lower precision, but at the same time, we are able to recover over 50% of the psoriatic arthritis. So I really like one of the points that we all mentioned about, like predictions also have to be dynamic. So this is using a genetic data alone. So we do believe that with the integration of more dynamic data like the genetic data, we're able to increase the performance for this kind of PSA assessments. So in the last part of the talk, I'm just going to show you some recent work that we have not been published, which is a work that is supported by Amgen. They're trying to use genomic data to assess the drug response. So in a recent work, we tried to look at using the cytokine stimulated keratinocyte signatures to really characterize different skin types, including including unevolved psoriatic skin or unevolved atopic dermatitis skins, and also try to look at the difference between them. Okay. And then at the same time, we also try to apply machine learning approach. This is like a self-organizing map, like clustering approach, the nonlinear clustering using the existing psoriatic lesional skin data that we have to try to look at the potential heterogeneity among the lesional skin of psoriatic patients. So all this kind of like pop up to say, well, some of those in vitro experiments that we obtained, that again, so that's different cytoplasmic keratinocytes signatures could really help us to make some biological inference. But how about, can we actually apply it to see whether we could provide whether this could be good features, informative features for us to provide drug response assessment. So we look at our RNA-seq data for patients that undergo anti-TNF response. So we look at the baseline expressions for those individuals in the uninvolved skin or the psoriatic skin. Okay, and then we identify those genes that show the strongest associations. So in the baseline versus those in the subsequent week PASI improvements, and then see whether they are actually enriched among the cytokine signatures that we identify in independent in vitro RNA-seq data sets. So indeed, we actually find that the uninvolved skin, but not the additional skin, have the baseline expression levels that are predictive to future PASI changes that are enriched among the interferon or the TNF signatures. So we use this as informative features. So these are just two examples here, two genes examples here. So the x-axis representing the baseline expression levels for that gene. Okay, for example, this is like KLT2 with you seeing a low expression for this gene in baseline, then we would expect the weak trials to have high response. And the example is like the ES15 genes. If you see a high expression in the baseline, we would expect that to have a higher changes in the PASI by week 12. So again, we're using this to really try to assess the drug response. Okay, we try to look at different models. The top, this is the light blue line here representing the best model that we identified. It reads as for the top 20% of patients that we predict to have they achieve the PASI 75. We are able to get around 80% in accuracy for predicting that. So this is again, this is trying to integrate multiple information, the in vitro experiments that we have with the experiments that we get in the baseline, molecular profiles that we got in the baseline, and also the clinical response PASI in the subsequent weeks to achieve this. So I do want to try to say how the best ways to really address the challenges that I mentioned. Again, sample size is key, not only for a better estimation for the parameters that, for example, in the case for the modeling the gene expression levels using genetic variations, we show that with the larger expression panels, that we are able to achieve better results or more robust estimations. And then also increasing sample size will help us to really avoid overfitting by allowing the cross-validations, leaving some out for the future testing while using all the other rest for model training or optimization. And then, reproducibility is always an issue. We always see that, for when applying some of those machine learning models, especially really good if we are doing training on particular institute or a hospital, but we apply a model that's trained in one hospital but and apply to the others, then we got loss of some performance. So I think this we have to account for the multiple different factors in here, and also I think we have psoriasis has been studied for a lot, like the genomics data and the genetic data, but I think a lot of those are still missing some of those clinical responses. So if we could also link that to the existing or the to be collected molecular data, would be really nice. And then this is going to be really useful. In addition to only relying on one or two techniques, trying to integrate multiple different omics to really make the inference would significantly improve the result. So in here, this column is just to illustrate this idea. So assuming that you have like multiple different data sets, so each data set could have a different types of features. So we do not want to wait until we're only using the data set that have all the features that I need to do the data integration. But at the same time, if we could leverage some data set that could be, well, missing some, but have the others to make a more refined model, then we should take advantage of that. And this could happen, when we are integrating multiple different computational biology approach to achieve that. So with that, I would like to thank all my colleagues and funding resources, and we'll be happy to talk about